Dear learners, welcome back to NPTEL online certification course on novel drug delivery systems. I am Dr. Satish Devanapelli, Assistant Professor at Department of Pharmaceutical Sciences and Technology, Institute of Chemical Technology, Mumbai. We are discussing on the topic of protein peptide and nucleic acid delivery systems. In our third session, we are going to discuss on the various approaches for protein peptide delivery systems. So far, in our two lectures, we have discussed on the introduction on protein peptide drug delivery systems. In our recent lecture, we have covered on the key considerations of protein peptide drug delivery systems. So, today I am going to discuss on the other topic on the barriers to protein peptide drug delivery. So, in somewhat we have learned during the introduction and the previous session about some of the limitations, or you can say during the key consideration, and also in the general discussion we learned on the few of the factors, but today we are going to discuss very detailed on the barriers to protein peptide drug delivery. So, yes, we know that delivering protein peptide effectively is one of the major challenge because of the so many limitations from the intact as in a protein, like a stability, storage and degradation, and then formulation also one of the major challenge. During the process, selection of the excipient is also one of the major challenge, and after developing stability also one of the major problem, and after delivering into the body, so again we will face lot of the problems. So, which we can classify into the like protein itself, like they have the problems in the native structure, and also during the formulation, and with the formulation we have lot of issues, and in the delivery also we have problem, and the fourth one is the in vivo in the body, or you can say in biological pH or biological conditions also will face lot of issues. So, which ultimately affect the absorption, bioavailability and the efficacy of the molecules. So, these are can be classified into biological and physiological obstacles. These are hurdles we need to overcome by designing of a novel formulations. So, that is why we are learning key consideration about the physicochemical properties, as well as now we are going to see the barriers for the protein peptide drug delivery. So, yes, first one comes the physiological barriers, and the second one, whenever you want to go for the oral, the intestinal epithelial barriers, and third one is the capillary endothelial barriers, and the fourth one is blood brain barrier. So, here we are going to discuss the within the body what type of barriers we are facing in developing the protein peptide formulations. Now, the first topic is physiological barriers. As we have discussed very well that enzymatic degradation is one of the major concern in the physiological barriers. These are the enzymes, proteolytic enzymes, pepsin, trypsin, chymotrypsin, which will degrade the proteins before absorption, right, as we know. So, proteins are the building blocks of the amino acids, and yes, with the polypeptide chain they will connect each other and it will form a protein molecule, right. So, these enzymes will degrade the proteins into the peptides, small peptides as well as amino acids. Once it is processed, then that leads to the inactivity of these biological molecules. So, the one of the physiological barrier is that enzymatic degradation will happen within the body at different biological target site. And the second one is again in the physiology, the pH, as we learned in our previous session, right. The oral, the stomach has a 1 to 3 pH and intestine has a 5 to 8 and the blood is plasma, pH is 7.4, ocular 7.4, and whenever you want to go for oral, that of course comes whenever you want to give in the oral delivery, mouth or oral disintegration type of formulation, you need to consider pH 6.8 to 7. So, GI tract has variable pH which can denature the proteins, right. So, that is also second factor. We will see with the picture representation in our next slide. And third one is mucus barrier. Yes, thick mucosal layer traps and removes the protein via mucosal turnover. This we have learned in the mucosal drug delivery system, right. So, mucociliary clearance, right. So, with that also we will face a problem in the absorption as well as bioavailability and therapeutic efficacy. So, we need to understand the pH related studies with the proteins and also the mucosal barrier. We need to, for example, if you are looking for a nasal delivery or if you are using for a buccal delivery and the ophthalmic and even pulmonary, we should know the what is the turnover, or we can try to understand the mucociliary clearance or the barriers of the mucus at that particular site. And of course, it not only happens with the physiological pH, the change may happen with the even with the formulation also. So, that we need to understand to overcome for that required formulations. So, inter-individual variability, so that is related to the patient concerns, even the GI physiology also varies, the enzymatic levels also varies, and the pH, the motility effect for that will affect the absorption. So, this inter-individual variability also comes as a major barrier in the physiology. Now, coming to this, you can see here the picture representation. So, this is the digestive tract. Now, here you can see the physicochemical barriers. You can see the stomach and its pH is 1 to 3, pepsin, lipase, and the surface area is very small. And come to the small intestine, pH 6.5, protease and bile salts are available, and the large intestine pH 5.7.4, microflora also, that is also will affect the problem. And now coming to the second one is a cellular barrier. Now you can see the tight junctions, also various cells present, so that will also hinder the release or permeation of the molecules. And as we discussed mucosal barriers, now here you can see the mucin. So, this is a mucin layer and the particles, whatever the molecules which are trapped in the mucus, so which cannot permeate. Yes. So, with this the various types, like whatever we have seen, the topics which are enzyme degradation, pH gradient, mucus barrier, inter-individual variability also comes under the physiological barriers. Now, coming to the second barrier, that is intestinal epithelial barrier. Already we discussed on the pH in the intestinal variation. Now, this contains various types of the cells which will also works as a barrier for the delivery of the proteins and peptides. And as we learned in our previous session about the key consideration, if the molecule is greater than 500, which comes under the large molecular weight, which will the major limitation that such type of molecule will not follow transcellular transport. So, so that is also one of the limitation which comes with the intestinal barrier. Now, paracellular transport is blocked by tight junctions because of their pore size is very less. Again, so that is one of the major challenge. So, whenever you are delivering the molecule and you need to understand what are the intestinal epithelial barriers based on their requirements. And the another one is M cells which assist in the limited uptake of the macromolecules. So, that is also we need to consider while developing the protein formulations. So, far we have seen physiological barriers and the intestinal epithelial barriers. Now, third one is the capillary endothelial barrier. So, endothelial cell layers of the blood and lymph capillaries prevent the diffusion of the large molecules because of their pore size as well as the restricted layers which are presented in the capillary endothelium. So, here now you can see the pore size, it is very small, 6.5 nanometer, too small for a proteins, right. So, such cases we have to come up with the nano formulations, so that will help to penetrate from this pore size. So, albumin, other plasma proteins are restricted to the vascular compartment. Now, you can see these are the endothelial cells and these are the pericytes, and here is the pericyte nucleus and also endothelial cell nucleus. So, to overcome this we should come up with the carriers or some cell penetrating peptides. And another one is the blood brain barrier. So, whenever you want to target any type of neurodegenerative diseases or central nervous systems, such cases, and if you want to pass or if you want to reach the molecule into the brain, you have to pass the tight junctions which are presented in the brain. So, you need to come up with the right perspective, like with the functionalized nanocarriers and the liposomes, carrier systems, cell penetrating peptides will help to overcome all the challenges which are there in the blood brain barrier, which is a highly selective barrier that blocks most large and hydrophilic molecules including proteins. Okay. So, yes, tight junctions and active transport systems will limit the CNS access, and now you can see the molecular cutoff, it is very less, very few proteins have this much range, 400 to 500 Daltons. Okay. Right. Yes, there are the strategies. With that you can use even intranasal delivery systems by using of various formulations and spray you can avoid. Now, you can see here this is the capillary and these are the astrocytes, and now you can see this is a blood brain barrier. It is represented in the picture representation. Now, you can see, so these are the tight junctions, so which will not allow the drug to reach to the blood. Okay. So, yes, there are the agents which will also stop the function of the tight junctions, which will change the structural modification of the tight junctions, which will allow to reach the drug molecule into the blood. Okay. So, even by using of the intranasal delivery, we can also bypass this blood brain barrier which will reach to the brain. the another topic, that is the techniques for stabilization of the proteins and peptides. So far we discussed instability, and as well as we need to focus on how to stabilize the proteins and peptides. So, that is also important parameter in the protein peptide drug delivery systems. So, the first one comes with the conformational stabilization, the colloidal stability, stabilization by drying, and the chemical modification. We will see one by one. So, first one is in the conformational stabilization. Yes, you can do the stabilize the molecule by using of the buffers and the surfactant. This already we discussed in instability issues. By changing of the pH, it leads to the loss its activity, it undergo the degradation and instability issues. So, we need, by providing the buffers, we can resist the pH change and maintain the protein conformation as it is with the native. Some buffers directly bind to the proteins, like phosphate binds to the fibroblast growth factor, which will stabilize. So, these are the common example of the buffers which will maintain the pH. Phosphate buffer, tris buffer and citrate, these are most commonly used while developing the biological formulations. And surfactants, so we use polysorbate 20 and 80 as a polyethylene glycol, even pluronics we use as a surfactant, which will reduce the interfacial tension between the aqueous and also hydrophobic regions of the proteins and peptides, and that also suppress the aggression. These are the most commonly used the surfactants during the formulation that will stabilize the formulation, and this will work as an aid in refolding of denatured proteins and reducing the surface adsorption. So, to conformational stabilization we can use buffers as well as surfactants. We can also use polymers. So, here the polyethylene glycol, the dextran and gelatin, we can use it as in a conformation stabilization. The people use the polymer drug conjugation or polymer peptide conjugation is the best strategy. So, with that we can improve the conformation stability of the molecule. Metal ions, yes, zinc and magnesium and ferric, so which will also help in the stabilization of the proteins by binding directly to the at metal side. So, here is the example, calcium is stabilizes the DNAs. So, similarly we can use various types of the metals which have the conformation stabilization property. Yes, you need to select right metal ion, that is one of the major issue, and after that we need to understand the right concentration of metal ions for the stabilization of the proteins. So, the another topic, that is colloidal stability. So, here you can see in the picture representation. So, we will see these are the common problems in the biological molecules, like aggregation, or you can say separation, settlement, nothing but sedimentation of the protein molecules or biological molecules is one of the major problem during the storage, right. So, we can use various types of the even formulations by modifying the viscosity and by changing the pH, right. So, by making a like a colloidal formulations also we can improve the colloidal stability of the formulation. Now, you can see the protein excipient interaction will affect the solubility, viscosity and also aggregation. So, colloidal stability is essential to prevent unwanted aggregation and crystallization. by using of crystal growth inhibitors as well as viscosity modifiers, solubility enhancers, and by prevention of the aggregation by using of surfactant as well as the colloidal systems, we can maintain the colloidal stability of the formulations. And the another one is stabilization by drying. Now, whenever drying comes, we need to think about the water, right. So, we need to remove the water from the formulation. Water is considered as a good raw material in the pharmaceutical industry, but similarly it has a disadvantages. We use water as a for the solubility medium, and not only that, we use it as a vehicle whenever you want to give the injections or even for the oral liquids, right, even some of the suspensions and in the emulsions, right. And not only that, when you want to go for the sprays, definitely we have to use the water, right. So, water can be used as in a dispersing medium, also we use as a solubilizing medium, even for the medium for the dissolution studies also. We use water like anything in the pharmaceutical industry based on the requirement. Even for the parentals also we use water for injection, we do not use purified water, right. But this water also has one of the disadvantage with some of the product, nothing but. So, water will create the microbial growth in your formulation, instability issue, nothing but hydrolysis one of the major problem, right. So, to avoid this, we need to remove the water from your formulation. Yes, drying can be possible, but all drying techniques are not useful for the biological formulations, because whenever drying comes, we need to remove the water from the formulation by heating, right. So, freeze drying is a technique, it applies sublimation, nothing but the ice will convert into the vapor phase without converting into the liquid medium. So, which is called as sublimation, also known as lyophilization, which will enhance the shelf life, thermal stability, and also ease of the transport. Definitely 1 liter of formulation which consider containing of 10 gram of product, right. The total weight will become near 1 kg. When you are removing the water and your formulation, you will get a 10 gram. With that, we can do easy transportation, you do not need to use the proper protection during the transportation. So, risk during the freezing phases, that is a denaturation, aggregation also happens. For that, we need to use some of the cryoprotectants or lyoprotectants. You can also say, yes, it is already mentioned here, sucrose, trehalose and maltose, which can reduce the all type of the problems we face during the freezing as well as drying, right. So, with that you can make your products into the dry powder, yes, but all techniques are not feasible, like spray drying. If your product is, if your protein peptide is stable, you can use spray drying, also field drying also possible, and vacuum drying also possible. So, the last one is the chemical modification. So, here we will need to modify, as we discussed, the deamidation happen in the instability issue. For that, we need to use the amino acid modification, we need to alter the side chains by using glycosylation sites. Here is the one example, vasopressin analogues, that is nothing but desmopressin. Here the D-arginine substitution will improve the stability and absorption of the vasopressin analog. So, we need to modify, and second one is hydrophobicity. By adding of hydrophobic groups, we can improve the membrane permeability and also enzymatic resistance. We can also improve the oral absorption of the insulins. So, here is the best example which I already mentioned, right. We can use hydrophobic groups, which will improve the lipophilicity as well as mucosal membrane. It will improve the stability against the enzymatic degradation. Pegylation, we already discussed this very clearly in the liposomals, like pegylated liposomes. When attaching with the PEG molecule, what happens? Your RES system will decrease and half life also will increase, solubility will also increase. Not only that, that will inhibit the all type of other issues, proteolysis, renal clearance, immune response, which are related to the reticuloendothelial system. With that, but there are the drawbacks, activity due to steric hindrance and also leads to aggregation. Now, you can see these are two example proteins and peptides. They showed the improvement in the bioavailability as well as efficacy. Now, hyperglycosylation. So, here we add polysaccharide groups directly to the protein or peptide to mask the hydrophobic region to balance the hydrophilic lipophilic balance, which will improve the solubility, reduce the immunogenicity as well as circulation time. So, the glycolization is one of the important topic in the protein peptide and even the vaccine delivery. So, this will decreases the steric endurance. So, mannose-ylation, when you want to target the mannose receptors, and you can use mannose ligand and we can bind and we can use it for the various types of the infection targeting. In such case also we can use the targeting of the liver. So, density of mannose will affect the recognition efficacy in the formulation, and these are some of the important components we can use in the protein and peptides directly and indirectly. So, that is for pH. pH, it also avoid the all type of the degradation. Stabilizers we can also use, these are the surfactants, sugars and salts can be used. Solubilizing agents, that will improve the solubility and they should work effective at low concentration, and the buffers that will maintain the pH, and phosphate, acetate, histidine, glutamate and other buffers. Tonicity modifiers. So, when you are go for the parenteral, we can use sodium chloride, that is 0.9%, and you can also use 5% dextrose, sorbitol, mannitol, glycine, which will maintain the isotonicity to the blood pH, and bulking agents that will also improve the total weight of the product. So far we discussed on the various barriers and the important components used in the protein peptide formulation. So, in our upcoming class we are going to discuss on the some of the formulation considerations and the delivery systems from the different routes like parenterals, oral and other non-invasive routes. Thank you so much. See you soon.